Hey everyone! Today we're going to talk about the tuning curve, which is one of the most fundamental tools people use to talk about what different neurons do. And so remember, neurons are the cells that make up the nervous system. And in fact, there are about 100 billion neurons in the human nervous system, and they're connected together into a massive network by these connections called synapses. And what's really amazing is that somehow through this massive connectedness, the nervous system allows us to move around, to have sensations, to think, to imagine, and to do basically all of the things that make us human. And if you want to know more about the anatomy, you should definitely check out some of the other Khan Academy videos on the nervous system. The main question we're interested in today, though, is, what are these neurons doing? Like, if I were to pick this neuron here, for instance, sure, I can talk about it, sodium and potassium and myelin and all that, but how would I talk about what its role is in making the nervous system work? And to answer this, we're going to use what's called a tuning curve, which is really just a nice graphical way of describing what a single neuron does. All right, but before we get to how tuning curves are actually made, we have to first remind ourselves what makes neurons so special as compared to, say, liver cells or kidney cells. And the answer is that if conditions are just right, neurons will emit action potentials, or spikes, which are these quick little electrical signals. So if you were to listen to an active neuron on a speaker, for instance, maybe this guy over here, you'd hear these little clicks going by. And those are action potentials. And so it's widely thought that spikes basically constitute the language that neurons speak. This allows us to now formulate our question a little more precisely. We can now say, what do a neuron's spikes mean? What information do they convey about the outside world or our internal state? Okay, so a pretty common experiment used to ask this kind of question looks something like this. So there's a mouse, maybe. That's my mouse. It's got a little tail. And there's an electrode recording spikes, so remember spikes, from a neuron in, say, the visual region of the mouse's cerebral cortex. So maybe spikes from visual cortex. And the electrode is probably plugged into a computer so that you can record when the spikes arrive. In front of the mouse, there's another computer that is programmed to display different images or different stimuli at different times. And the goal really is to understand in some sort of systematic way how the stimuli presented to the mouse give rise to different numbers of spikes in the neuron we're recording from. So the main trick to doing this is to choose not just images from Facebook or whatever to present, but to choose some specific set of stimulus images that vary according to some parameter. So in the visual cortex, for instance, it's generally thought that neurons respond to high contrast lines or bars at different angles on the screen. So you might have one that looks like this, or one that looks like that, and these would get shown to the mouse. And the parameter that describes these different stimuli is simply the angle, or the orientation of the bar. And so it's quite simple. The images get shown to the mouse for, say, 2 seconds at a time, and the spikes from the neuron get recorded by the electrode and stored on our computer. Okay, and so this is science, so we'll be systematic. So we'll make a table of all the different stimuli that get presented. So maybe this guy is at 0 degrees, this guy is at 30 degrees, this guy is at 60 degrees, 90 degrees, and so on and so forth. Actually, we don't need this last one because it's really the same as 0 degrees. And people usually present the stimuli in a random order. So maybe this bar gets shown first, the 90-degree bar, and the neuron spikes 2 times. So 2 spikes. Then maybe this bar gets shown, the 30-degree bar, and the neuron spikes 1 time. Then maybe the 150-degree bar gets shown, and the neuron spikes 12 times in those 2 seconds. And so on and so forth. So maybe we get something that looks like that. And then to be rigorous, the procedure usually gets repeated over many trials. So we'll call this trial 1, then we'll do trial 2, and again, maybe they're presented in a random order. So maybe we get 2 spikes for 30 degrees this time, 2 spikes for 0 degrees, 0 spikes for 60 degrees, maybe 13 spikes for 150 degrees, 4 spikes for 120 degrees, and 3 spikes for 90 degrees. And you can repeat this over and over again until you've performed all the trials that you want. N trials. And you fill in all of the spike counts and store them on your computer. Or maybe someone else has already done this and you're looking at the data set and want to make a tuning curve for one of your classes. And so a big table is a nice way to store things on a computer, but it would be even better to have a graphical representation. And so what we can do is to make a little plot. So on the x-axis, we'll have the orientation that was varied. So maybe this is 0 degrees, this is 90 degrees. And remember, these correspond to different stimulus angles. And on the y-axis, we'll have the number of spikes the neuron emitted in the 2 seconds of our recording. And now in order to actually make our plot, the first thing we're going to do is to calculate the mean spike number and the standard deviation for each stimulus. So for the 0 degree stimulus, maybe we have a mean of 2.1 spikes and a standard deviation of 1.3. For the 30 degree stimulus, maybe we have a mean of 1.5 spikes and a standard deviation of 0.6 spikes, and so on and so forth. Right, so now we have a mean and standard deviation of the spike count elicited by each of our different stimuli. So now we can go ahead and plot those. And we'll go ahead and plot the mean and standard deviation for each stimulus. So for 0 degrees, it was 2.1 with a standard deviation of 1.3, which we'll draw as little error bars. For 30 degrees, it was 1.5 with a standard deviation of 0.6. Those are our error bars, and so on and so forth for all of the different orientations. And in fact, on this plot, we'll replot 0, which is the same as 180 degrees. Oops, this one should have been angled there. We'll replot 0 at 180 degrees, and then we just connect the dots. And so this is a tuning curve. It has a higher value for a stimuli that the neuron responded strongly to, and a lower value for stimuli that the neuron responded weakly to. And what's really great is that you can just look at a plot like this and instantly say, oh, this neuron responds strongly to bars oriented at 150 degrees. That's great. And then maybe this is another tuning curve for a different neuron that responded most strongly to bars oriented at 60 degrees. Or maybe you have another tuning curve for another neuron that looks something like this. And so this neuron also responds most strongly to bars oriented at 60 degrees, but it's not as sharply tuned. So it has broader tuning, since it still produces a sizable response to bars at, say, 30 or 90 degrees. Or you might have a tuning curve for a neuron that has a similar mean, but a much greater standard deviation. And so this neuron would still prefer orientations at 60 degrees, but it would be a lot more variable in its response. And so maybe that variability comes from uncontrolled inputs from the other neurons it's connected to. And so what's cool about tuning curves is that they're ubiquitous in neuroscience. They're used all over the place. So for example, you could have a tuning curve for a neuron in the auditory part of the brain, and maybe instead of the orientation of a bar, the pitch of a tone was varied. And maybe the tuning curve looks like this. And so this tuning curve would describe a neuron that prefers a 1 kilohertz stimulus. Other examples of quantities neurons have found to be tuned to in the brain are specific muscle movements, the location of an animal in a room, or specific textures that an animal is touching. And so tuning curves are really just these concise little graphical pictures that have a lot of information about what a neuron is doing, and they allow us to not really worry so much about the complicated network that surrounds it. And this is what makes them one of the most common and fundamental tools in neuroscience.